Hi everyone! Today we're talking about the cell membrane and cellular transport. This is going to be on page 23 of your purple packet, so go ahead and flip there now if you're not already there. So, all membranes are going to have a cell membrane, and they're the same basic structure across the board. They're all going to have a double layer of lipids and some proteins floating along in there. The lipids are responsible for the isolating functions, so that means that the lipids are really what's separating the inside of the cell from the outside. Proteins are going to regulate what's allowed to come in and what's allowed to go out, and it's also going to be important for cell communication. The cell membrane is known as a phospholipid bilayer, which means that there are two separate layers of phospholipids. The membrane is described as selectively permeable. Go ahead and talk to your neighbor about what that means. You might remember from the reading that this means only certain things can pass through. The cell membrane is really going to be the deciding factor of what can and can't get through into and out of the cell. Go ahead and jot down a couple of words on your paper and draw a picture to help you remember this idea. So let's take a closer look at these phospholipids that the cell membrane is made of. This is a diagram of what it looks like, and it kind of reminds me of a balloon with two strings instead of just one. So the top circle is what we call a phosphate head, and it is hydrophilic, which means it loves water. The fatty acid tails are hydrophobic, which means they're scared of water. This allows us to have this really cool membrane structure. So if you look right here, you have a phosphate head, fatty acid tails, and then the next layer is actually flipped. So we have our fatty acid tails and then our phosphate heads. This is really important because the extracellular fluid and the cytoplasm are both water-based. So it makes sense that our hydrophilic or water-loving heads are facing water on both the outside and the inside of the cell, and our hydrophobic tails are protected from the water. When we think of solutions, there are two main pieces to a solution. There's the solute and the solvent. Now, I don't recommend this, but if you were to rob a bank and you needed somewhere to put your loot, you could definitely stash it in the vent and use that as your escape route. Again, not saying I recommend that you rob a bank, but this is a great way to remember the parts of a solution. You put the loot in the vent. So your solute is whatever you're dissolving. So this might be sugar, salt, anything along those lines. And then the solvent is the water, usually, or whatever liquid is doing the dissolving. There are two types of cellular transport. There's passive transport and active transport. Passive transport is when molecules are moving with the concentration gradient. So that means they're going from where there's a high concentration of those molecules to where there's a low concentration. This process does not require energy. Here's a couple of examples of passive transport in action. You can see that there's a higher concentration of the pink molecules on this side and a lower concentration here. And they're going from high to low, and there's no energy being involved. Same thing is happening over here in this diagram. We're moving from high to low. Because we're moving from high to low in both of these examples, these are both types of passive transport. Go ahead and talk to your neighbor really quickly about these 4 questions. Just to make sure that we all got those answers right, number 1 is the cell membrane. Number 2 are phospholipids and proteins. Number 3 is passive and active transport. And number 4, molecules are moving from high concentration to low concentration without the use of energy. Here are 3 examples of passive transport. Diffusion is the movement of solute molecules from an area of high concentration to low. Facilitated diffusion is the movement of solutes through channels from high concentration to low, and osmosis is the movement of water from high concentration to low. Since they're all passive transport, they're all going to be from high to low concentration, and none of them are going to require energy. Here you can see that movement from an area of high concentration to low as those molecules spread out. Here's another example. In the top left corner, we can see that we've got a ton of these orange dye molecules on the left and none of them on the right. They're going to diffuse across that membrane until they achieve equilibrium. And this is where we have an equal concentration of molecules on either side. It's really important to notice the size of these molecules and the size of the gaps in the membrane they can pass through because they can physically fit. The bottom diagram is showing us how we have two different molecules, and each of them are going to move according to their own gradient. So it's not enough to just have a certain concentration of molecules in general. The maroon and the orange each want to be in equilibrium. So we want to have the same concentration of orange on each side and the same concentration of maroon on each side. Another really important thing to remember is that we're not actually interested in the number of molecules, we're more focused on the concentration. So in this case, we have a concentrated dye on this side and a not so concentrated dye on the other. This membrane is selectively permeable, and it's not going to let these dye molecules through because they're too big. Now, instead of relying on the dye molecules to move through to achieve equilibrium, we can actually have the water move through. And now, even though we still have the same amount of molecules on this side, the concentration is lower, and the concentration on both sides is equal. Here we see diffusion happening. We're going from an area of high concentration to low, and we're able to just pass through that phospholipid bilayer. This is facilitated diffusion. We're still going from high to low, but we're having to use those channel proteins to help us move through the membrane. And last but not least, this is an example of osmosis. You can recognize that this is a water molecule by its Mickey Mouse shape. Remember that water is H2O, so here are our two hydrogens as the ears, and then the face is the oxygen. Go ahead and talk to your neighbor about what's happening in this diagram. Here's another 4 questions to talk to your neighbor about. Diffusion is the movement of solute molecules from an area of high concentration to low. Osmosis is the movement of water molecules from high concentration to low. Facilitated diffusion is the same thing as diffusion, but this time we're using these channel proteins, and they're going to create a passageway for molecules to move from one side of the membrane to the other. All of these processes occur because molecules want to spread out and have an equal concentration. Let's take a look at some specific examples of osmosis. Here we have a cell that's 80% water, and it's in an environment that's 100% water. Remember that water is going to move from an area of higher concentration to lower concentration, so that means the water is going to move into the cell. That's going to cause our cell to swell, and it might even burst. The next example, we have 80% water inside the cell and 80% water outside of the cell. Both areas have an equal concentration of water, so water is going to move in and out at the same rate. There's no net movement of water, so this cell is going to stay the same size. Our last example, we still have our 80% water cell. This time the environment is 70% water. Water is going to move from the higher water concentration to the lower, so it's going to leave the cell. This is going to result in our cell shriveling up and getting a whole lot smaller. Well, that's cool and all, but why do I even care? How does passive transport affect my cells? Here's a great example showing how red blood cells are affected by their environment. In this middle situation, we have the same concentration of solute on the inside of the cell and the outside. This means that the water is going to move in and out of the cell at the same rate, and there's no net change. Our cell's not going to grow or shrink, and life is good. On the left, we have more solute on the inside of the cell than on the outside. Because of this, the water is going to rush into the cell, and our cell's going to swell, and it might even burst. On the other hand, this solution has a lot more solute than the cell does. So we're going to see that the water is moving out of the cell to help dilute this environment. That results in our cell shrinking and shriveling, and it won't be able to carry oxygen anymore. So we can sum up the importance of cellular transport in one big word: homeostasis. Cellular transport is going to help make sure that things that need to come into the cell can come into the cell, and things that need to leave the cell can leave the cell. It's going to help us maintain homeostasis. Now, we need some fancy sciencey words to help describe these solutions. This is on the chart on page 25. You've got a space for definitions on this chart, so go ahead and jot these down. Right now, in this U-shaped tube, I have a lot more solute molecules on this side. This side is more highly concentrated, and so we're going to call this side hypertonic. I like to think of hyper as rhyming with higher. We all know kids who get really hyper and they have a lot of energy, so they have a higher amount of energy than normal. The left side, on the other hand, has less solute than the right, and so we can describe the left side as hypotonic. Remember, hypo rhymes with low, so it has a lower solute concentration. Now, the water molecules are going to scooch over to achieve equilibrium, so now we have an equal concentration on both sides. We call that isotonic. Isotonic means there's an equal concentration on both sides of the membrane. I went ahead and switched over to the DocCam just so we can all fill in this chart together. You should already have these definitions filled in. So, if I take a beaker and I put an animal cell in it, and my solution is hypertonic, that means that I have a lot more solute on the outside than on the inside. Water is going to move out of the cell to help dilute that higher concentration in the environment, and my cell is going to shrivel and shrink. That's not good for the cell. A plant cell is going to do the exact same thing in a hypertonic solution, but remember that a plant cell has a cell wall. So our cell is going to look a little something like this, and the membrane is actually going to pull away from the wall because the cell itself is shrinking. This is a really interesting process called plasmolysis. We can call this cell plasmolysed. On the other hand, if we put an animal cell in a hypotonic solution, let's go ahead and see what that would look like. So we've got a beaker of water, we've got our cell. The solution is hypotonic, which means that there's a lot more solute on the inside than on the outside. That means water is going to rush into the cell to try to dilute the cell and get an equal concentration on the inside and outside. This is going to cause our cell to burst or lyse. Lyse means to break, like a lysosome breaks things down. If we place a plant cell in a hypotonic solution, it's going to do the same thing. Water's still going to come in, but remember that cell wall? This is actually going to protect the cell from bursting. So we're going to see pressure building up against the cell wall, but the cell wall is going to prevent it from bursting. This is actually the happy place for plant cells. This pressure against the cell wall is going to help them remain upright and not wilt. An isotonic solution means there's the same concentration of solute on the inside and outside of the cell. That means that water is going to be moving in and out at the same rate, and this is actually the happy place for animal cells. If a plant cell is placed in an isotonic solution, there's going to be an equal amount of water coming in and coming out, but we're not going to have this pressure building against the cell wall, and this is going to cause our plant cells to begin to wilt.